So in the final lesson of this unit, you will have been looking at drug trials and how scientists go about testing and developing new drugs to make sure that they are safe for human usage. So there are two key terms to make sure that you are happy with. The first one is preclinical trials. There's said here. Now, this is the first stage that a developed drug goes through, and that is testing on cells and tissues in a lab to identify any damage to individual cells that it might cause as an unintended consequence. The second stage is then testing on live animals, and that is to test specifically for the efficacy of the drug, so whether it works particularly well and produces the required effect, and also to test the toxicity of the drug, so to test whether it's harmful or not. And so they're basically trying to identify the best amount of the drug to use as a dosage. In the UK, for example, it's the law that new drug needs to be tested on 2 different animals. Obviously, there are ethical elements that come into consideration in this, but importantly for you, it's about knowing the stages. Now, the next stage then is what's referred to as clinical trials. Now, clinical trials, you might have guessed, involves testing drugs on humans. Now, obviously it only enters this stage if it passes through the preclinical trials, and then if we're talking about the different stages, first stage of this process is testing it on healthy individuals. Now you might ask why testing on a healthy individual. The reason being is that so any side effects can be identified, and also these aren't or not at risk of being confused with symptoms of an existing illness. And so the first dose is particularly low, and then it's gradually increased during the trial, and this is to find the optimum dosage, so the most effective with the least harmful side effects. Then, to ensure fair testing, volunteers are split into 2 groups. Some are given a placebo, which is a fake drug, and it looks like the drug, often made out of sugar, but doesn't actually have any effect on anything. A doctor who gives a patient a placebo can then assess if the drug makes a difference or whether the person has improved because of unintended psychological effects of thinking that they'd taken the drug. And that's what we refer to as the placebo effect. After that, when we're talking about the next stage, stage 2 of clinical trials, that's when patients who are suffering from the illness actually receive the drug. And that's obviously to test whether it works. And this stage of clinical trials is often referred to as a blind trial. So the patient doesn't know if they are taking the placebo or they're taking the actual drug. And you sometimes get double blind trials where the patient doesn't know and also the doctor doesn't know who's getting the drug, who's getting the placebo. So any data that's collected can be guaranteed to be unbiased. test and be a fair reflection of the impact of the drug. And then obviously after that final point, everything is peer reviewed, so findings are sent to other scientists, they review it, see whether there's errors or anything like that, and then after that we can eventually get to the point where the drug can be put into the market and prescribed, and that should be everything that you should need to know about drug trialing.